Hello, that's hello, Arsidas sir. Sir, can you hear me? Sir. কি সব উঠি দিচ্ছিস না কি সব করে দিচ্ছিস বলছি তো কি করতে পারবে না সেই করতে পারবে না
हाँ हाँ
হ্যাঁ
तो एलाउ कर लेना मोर अनम्यूट होची
হ্যাঁ অ্যালাউ করে
ते बहुत पार्टिसिपेंट कम होछने
হ্যাঁ only 14 participants still now. We'll wait for 5 minutes, sir.
हाँ but you inform ask Gaurav
Yeah, Gaurav is there. Change.
you wait maximum up to 20 then we will start hello
Yes, sir.
20 हिले start करिया आ wait कर दिया 20 हिले start करिया 20
Okay. The number of participants. Okay. Okay.
Koshi, I have a question.
Sir.
Acha, do you organize this FDP program in every Sunday or it is a regular day, every day?
Sir, actually, 4 colleges are organizing this.
Okay.
Each Sunday, that is our turn.
Okay.
Okay. Sir,
On
second
every
and third other colleges are also.
Every Sunday you are organizing this program?
No, sir. Not every Sunday. Every that is last Sunday of every month. Our college is organized, and on second Sunday and third Sunday, other colleges are organized.
Yes. That means, who are the participants mainly?
Sir, participants are the teachers, faculty members
of the different colleges in
of
Odisha.
Different colleges under BPUT, sir.
Under BPUT only.
Under BPUT.
Okay. Actually, I think BPUT is now controlling all the pharmacy college and the technology college, no?
Right, sir. Most of the college, because in our Berhampur University also pharmacy department is there. In Utkal also pharmacy department is there. But other all for private colleges that is under BPUT, sir.
Except a university department, all the private and also government college also, no?
Yes.
Government
Sir,
college?
we don't have any pharmacy government college in Odisha, sir.
Okay.
Ours is the oldest pharmacy college in Eastern India.
That I know. That
Yes.
I know from the very beginning.
Yeah.
I was in Odisha. In 94, 95 to 97 in at Jharpokhuria, Srimanto Institute of Pharmaceutical.
Jharpokhuria.
I was there for the 2 years. I stayed at Baripada.
Baripada, okay, sir.
Thereafter, I shifted in 97.
Sir, almost 19 participants are there,
Okay,
and I think within this time they will also join. So, can I
No
start,
problem.
sir?
Yes, it is.
Okay. Thank you, sir. Namaste and good morning to you all. This is Kaushik Sharkar, Associate Professor and the Coordinator of the BPP NCFDT program. Dear colleague, on behalf of the College of Pharmaceutical Sciences, Mohda Berhampur, I welcome all the pharmacy professionals to this signature event of the third BPUT Online Faculty Development Program 2022. The program has already shaped to be excellent with all your gracious presence and of course in the presence of our erudite speakers. Today's topic is the role of molecular design in drug therapeutics, and the eminent speaker is Professor Dr. Achinto Saha, a distinguished scientist in the field of CADD, computer-aided drug designing. Actually, I am honored to get an opportunity to learn molecular docking and pharmacophore mapping under his tutelage in the year 2012, date back to almost 10 years back at Calcutta University. It was really exciting and exhaustive. And the thing that I learned from there also, I think the same notion will be also passed by sir today also. So in this regard, I would like to request the Honorable Vice Principal of our college, Mr. R.C. Das sir, to introduce our distinguished guest speaker to the forum. Over to you, sir.
Okay, thank you, Kaushik. Hello, am I
Yes,
audible?
Yes.
Yes, sir.
Okay. Very good morning, esteemed BPUT officials and principals and dear faculty members. I, on behalf of BPUT Odisha and Nodal Center Faculty Development Training Program CPS Mohda, have the honor to welcome you all to this third Faculty Development Training Program in Pharmacy of BPUT organized by CPS Mohda on the topic Role of Molecular Design in Drug Therapeutics. Our speaker, Professor Dr. Achinto Saha. Honorable Vice Chancellor, Professor Dr. C.R. Tripathi, sir has introduced 3 programs. One is Faculty Development Training Program for faculty members, Career Counseling and Guidance for students, Industry Institute Interaction and Training Program for students, with the objective of continuous improvement of quality and standards of students, faculties, and institutes. Meanwhile, already 13 such programs are conducted successfully by 5 nodal centers, and 14th and 15th programs are scheduled today to be conducted by CPS Mohda and TPC Barpalli. We are thankful to our Vice Chancellor. I am also thankful to Dr. B.B. Mukherjee, Director Curriculum, and his team for timely processing and providing assistance in smooth conduct of the programs. I am thankful to our Dean, Professor Dr. S.K. Mahapatra sir, and the principals of pharmacy colleges of BPUT for their continuous support. I am also thankful to our principal, Professor Dr. Subrat Sen, who has the key role in successful conduct of such programs. I am also thankful to the faculty members who have joined in this Zoom program. I must thankful our Nodal Center Coordinator, Kaushik Sarkar, and his team members, Subhasis Misra, Gaurav Saha, and others, who actually facilitate and manage the entire program. I once again welcome you all to this Faculty Development and Training Program. Now, on behalf of you all, I welcome Professor Dr. Achintya Saha, the esteemed speaker of this faculty development and training program. I am very much thankful to you, sir, Professor Saha, for accepting our request and accommodating this talk in your busy schedule. Sir, now Dr. Achintya Saha is professor and ex-head of the Department Chemical Technology, University of Calcutta, and he is working in Division of Pharmaceuticals and Fine Chemical Technology in Department of Chemical Technology, University of Calcutta. He's a professor in Department of Chemical Technology, University of Calcutta since November 2012 till date. And he was the head of the department from May 2015 to April 2017. His research area is computer-aided drug design and ethnopharmacology, computer-aided drug design, 2D, 3D QSR studies, pharmacophore mapping, receptor-based docking, molecular dynamics, and de novo design. Ethnopharmacology, estrogenic and contraceptive activities, PPAR modulation for herbal drug resources. He did his postdoctoral at School of Pharmacy, University of North Carolina, USA, from April 2008 to December 2008. His research guidance score is 35.07. He did his B.Pharm, M.Pharm, and PhD from Jadavpur University. He has, under his guidance, already 19 scholars were awarded PhD, and 6 are now registered. MTech 16 and MD Ayurveda 6. He was awarded SARC Fellow for University Grant Commission for Dhaka University in the year 2018, awarded Boy Scout Fellow by the Department of Science Technology, Government of India in 2007-8, and awarded Fast Track Young Scientist for Science and Engineering Research by DST, Government of India in 2006. Member Board of Associate Editors, Analytical Chemistry in 2002 to 2007, Indian Journal of Chemical Society. He has the ability to organize 4 weeks orientation programs for the college and university teachers, and he has organized 6 such orientation programs under his coordinatorship. And he has handled 11 research projects funded by AICT, UGC, and DST, etc. And one of the most important research project it's currently going on is the UGC DRSI project, Small Molecular Drug Discovery and Delivery Design Against Infectious Diseases, and worth 1.41 crore is under his coordinatorship. And he is a member of professional organizations like Fellow Indian Chemical Society, Fellow Institute of Chemists, and Fellow Indian Society for Technical Education ISTE, Indian Pharmaceutical Association IPA, Indian Pharmacy Graduate Association IPGA, Science Congress, Indian Science Congress Association, Indian Society of Pharmacognosy, Indian Association for Cancer Research, Indian Institute of Chemical Engineers, Association for Pharmacy Teachers APTI, Association of Chemistry Teachers of India, International Society for Computational Biology. And he has 152 research articles publications to his credit, and citations of 1,895. Published abstracts 95, book chapter 5, and other articles 6. This is just a brief introduction of Achintya Saha, and he is a legend in the computer-aided drug design. And sir, we are proud to have you today among with us. I hope and wish the lecture and interaction will be more interesting, and he will certainly ignite the young minds towards the modern technology-based research and software oriented research and modern instruments and softwares used for research. So with this brief introduction of Dr. Shah, I am proud to be associated with him now. Now I request Professor Achunta Sahasar to deliver his lecture.
Thank you, sir. Thank you very much
Yes,
for
sir.
your brief and the big
No, sir.
presentation.
Just a
Thank
brief
you very
only.
much. Okay, thank you. Okay. Jai Jagannath. Actually,
Sir, we are making you host now.
okay,
Sir,
sure.
we are making you host now. Yes, sir. Now you can share, sir.
Okay. Is it visible now? Is it visible?
Yes, sir. Sir, it's visible, sir.
Okay. Jai Jagannath. Actually, this is the last the weekend. You have a great and auspicious festivals of Jagannath. I'm also very much fond of Jagannath. So I think you will enjoy at this weekend. So start with this program. I have decided to select this topic, that is the role of the molecular design in drug therapeutic. As the I come to know that from the Kaushik that most of the participants are from the pharmacy background, they are the pharmacy teachers in different institutes. So I think they have very much habituate knowledge about little bit, because some are the chemistry, pharmaceutical chemistry, or maybe the other subject, but almost they have this information about the computer aided drug design under the curriculum of the BPharm and also in the MPharm program. So I took this topic. Basically, it is a old wine in a new bottle. Same thing we discuss, but in a different aspect. Just go through this one, and I think you can enjoy this program. Okay. Just a minute. I have. Okay. Drug discovery. I think you know about this map. It is a long road journey for the development of a new drug. That is starting from the exploratory development in the laboratory, passing through the synthesis, early stage study, that is we are talking about the preclinical study, formulation development, studies in the different clinical trial, phase 1, phase 2, phase 3, phase 4, and finally, if success, the drug has been go for the registration, NDA or MAA. So this is almost this one, but what are the steps behind this discovery? Identifying the disease first. Basically, it is the modern drug discovery phases. The scientists are now first identify what is the disease and what for we are going to develop the new drug. So after identification of the disease, we will go for identification of the causative factors, maybe the enzyme, maybe the protein. That may require at least 2 to 5 years. Then we will search for the small molecule, that is the chemical small molecule, which can modulate this abnormality or control this abnormality of that particular causative factor, that is maybe the protein or enzyme. That is also taking 2 to 5 years. Then go for the preclinical testing in the animal model, formulation design, and at the same time file IND, investigational new drug. Then go for the different phases in the human clinical trials, and finally, if it is approved, then it will come into the market. So it is a long journey starting from the exploration to the market. And this that is the average time is required almost 15 years. Cost, though it is mentioned 800 million, but basically now every day it is increasing and it is now comes to the 1.5 billion. So scientists, they are now thinking about the methods, approach for this drug discovery so that it can reduce the time. At the same time, it can also reduce the cost to bring the drug into the market. That's why first we will try to compare with the classical design versus rational design. And if you compare, we can come into to summarize that we have to go for first target identification. That is what is the causative factor for the disease. That causative factor first we have to identify it and properly validate it. Then go for the development of the small molecule that can lead to the generation of the lead molecule. But it is not so easy to optimize the lead. It will take another 4 to 5 years for this optimization of this lead. Then come for the preclinical and the final full development. That is the clinical test with different clinical phases you have to pass through. So if you start from the history, we cannot avoid any history. Basically, history is the giant leap of the mankind. Here also, you can see the discovery of the medicine or documentation of the medicine. We can see from the prehistoric Indus Valley civilization, that we can also say first Egyptian dynasty. What happens at that time? Some possible trial error based discovery, some Veda, some practitioner, they are supplying some medicine based on the natural system. Sometimes it cures, sometimes it does not. But knowledge is developing, subsequent development based knowledge is going on, and that can lead to the knowledge foundation. And finally, last 4 decades of the 20th century, when some information comes from this database, drug database, then we'll apprehend the quantitative relationship between chemical structure and the bioactivity. At that time we have that computer, supercomputers. Then we are shifting to this computer-aided drug design, and finally that we are now working on the molecular modeling and the cheminformatics. This study can help scientists to reduce the time at least 4 to 5 years and vis-a-vis the cost. What was the yesterday's drug discovery phase? People are looking for the natural compound. They are trying to isolate active ingredient from the natural source, from the different plants, different minerals. Then they are tested for the animal studies and formulation design, or after formulation design it is go for the clinic. But sometimes the people also started with the synthesis, pharmacology, and then clinical trial. But the chance of success was very poor. That's why people now they are thinking about the genomic study, because this type of advanced level studies are coming. So gene, protein, metabolomics, 3-dimensional structure, combinatorial chemistry, automated high throughput screening, virtual screening, docking scoring. These are the terms nowadays people are using to explore the drug more better way more scientific way that can also lead to reduce the time. So before I start, I just give you some basic information. What is the principal aim of a pharmaceutical technology, specifically as a pharmaceutical chemist? The main, it having the three roles. One is the discovery of the novel drugs with greater potential.